Earl, right?
At least two, maybe three.
The,
I just played a gig with Pavlov's dogs at the CNS meeting.
the, it sounds droolingly wonderful.
It was fun.
So for all of you, just to catch up, that Earl's the Pickauer Professor of Neuroscience at the Pickauer Institute for Learning and Memory at MIT. And he began his journey with Chuck Gross at Princeton in the inferior temporal cortex and face perception of visual features, and then did his postdoc with Bob Desimone, continuing that really important work. First clues also about memory and familiarity and things like that. But then, is it because of Pat you switched into prefrontal, Earl?
It is, absolutely.
So,
Pat taught me that prefrontal cortex is something you could study.
yeah, and because believe it or not, before then people didn't think you could.
Yeah.
And then I think we're all aware of just the fundamental, incredibly exciting data Earl has generated since, where he takes on, much like Pat, really fascinating questions on attention, categorization, and just gets beautiful data to address how these might happen with in vivo physiological recordings. He's also the provocateur of our field, who gets many of us hypertensive, and that serves to call attention to what we do, but also makes us think more stringently about how we think about it. So very valuable, and part of why we have you here today, because so important to exchange ideas. So thanks so much, Earl.
Awesome. Thank you. And thank you for the invitation. I really appreciate it. It's great to talk to you all. So I should ready to share my screen. Okay, start the slideshow. I said start the slideshow. There, so you guys should see my full screen now, right?
Yep.
Awesome. Okay, so let's dive right in. Hey, everybody, thanks for the invitation to speak. It's a great pleasure to speak to such a great group as you guys are. I'll just dive right in here. Our laboratory studies executive brain functions, much like many of you. And how we study them is much like many of you, we study them by recording electrical signals from the brains of animals, in our case, non-human primates. And we use a lot of computational modeling analytics to make sense of the signals. And we do so because we're interested in cognition and figuring out the brain. That's all well and cool, but we also understand cognition and dysfunction in diseases such as autism, schizophrenia, attention deficit disorder. It's one of the reasons why we're all given the grant money we're given. To conduct this research, we use fairly now called newish technology, multiple electrode recording. When I was a graduate student with Charlie Gross and postdoc of Bob Desimone, we used to record neurons from single neurons one at a time with a single electrode. This was due to technological limitations, but it was also due to the fact that, it's not that long ago where we first started to figure out how to actually read signals from neurons in the brain and the cortex. And, in order to get to where we are now, we had to figure out how the brain works in pieces at a time. We had to figure out the brain one piece at a time. And that's what we were focused on in the 20th century. Individual neurons, individual brain areas, what do they do? What's their function? And that was foundational, got us to where we are now. And where we are now is we can record signals from hundreds to millions of neurons simultaneously. And there's been the rise of computational analytics and modeling to make sense of these signals. And as a result, now in 21st century, neuroscience has sort of shifted in part. The studying individual neurons and individual brain areas is still very important, but now we've also shifted in part to understanding the brain on its network level, its emergent properties and mechanisms that emerge only when you see the parts interacting together. And I'll get to an example of what that is in a moment. But for now, what I'm going to talk about today is two different types of signals. which I'm sure you all are familiar with, spikes, which are these impulses from single neurons, think of them as individual voices of individual players in the brain, and LFPs, which are the average activity of millions of neurons, the roar of the crowd near each electrode. Okay? And we apply this technology to studying the prefrontal cortex and its role in executive functions. And I love this figure from Granger and Arnson, so I use it all the time. And what it says is that the prefrontal cortex is critical for planning, monitoring, and executing complex goal-directed behavior. Now, our hypothesis, and the hypothesis of others too, is that the major prefrontal cortex function is to learn and use the rules of the game. In order to engage in complex goal-directed behavior in the world in which we live in, you have to figure out how everything works. You have to figure out what goals are out there and what sort of behaviors are appropriate to reach those goals. And this requires acquiring and learning top-down information, knowledge of how the world works. We use that to predict possible goals and how to achieve them. Now, starting about 20 years ago or so, we began a series of investigations to test this hypothesis. We asked the question, does the prefrontal cortex really get tuned, absorb all this top-down information about how the world works? So first round of studies, we taught animals categories. We taught them the category of cats versus dogs, shape categories. Animals had to learn that one group of stimuli were arbitrarily designated to be a cat, and another group of stimuli were arbitrarily designated to be a dog. And there was a morphing space where we smoothly varied the shape between the different exemplars of the categories, but there was a boundary in the middle where we taught the animal anything on one side of the boundary was a cat, anything on the other side was a dog. The animals had to learn that. It was a boundary that did not exist in the real world, but the animals had to learn it, the top-down knowledge. We also taught animals, Andreas Nieder was in the laboratory, he taught animals the concepts of numbers 1 through 5. And much like the cat and dog study, the monkeys were trained to recognize these numbers regardless of how we presented them. It didn't make a difference whether they were 4 triangles or 4 stars or 4 pictures or 4 this or 4 that. The animals learned to recognize them as a number 4 and not pay attention to what the individual details of the display were. And we also taught the animals higher level concepts like same versus different, up versus down. And all these studies told us that the prefrontal cortex absorbs top-down information. When we record from prefrontal cortex neurons after this training or even during this training, we see the neurons evolve to represent this top-down information, and they throw away all the irrelevant details like what the display, which individual cat or dog, or what we're using to display number 4. They throw away that because prefrontal cortex doesn't care about that. They care about the prefrontal cortex cares about the rules and principles needed to solve the task at hand. So the rules of the game. And then we went on to show how the prefrontal cortex could use this top-down information to control the flow of activity across cortex. In other words, provide the control for executive control, to be a puppet master, to direct the cortex towards goals. And we showed that the prefrontal cortex kind of acts like an orchestra conductor. All this top-down information is like the prefrontal cortex learning the score, learning the big picture, and then using it to tell the rest of the cortex what to do to engage in that goal-directed action. Okay? Now, in conducting these studies, we found a property of neurons that we didn't know about in the 20th century. In the 20th century, we thought that neurons are relatively simple and specialized. Neurons were thought to detect single features like edges. And if you build up enough edges, eventually you get perception. Or simple combinations of features, like, for example, the red rolling moving ball here. All these things together equal the perception of the red rolling moving ball. That's the way we thought neurons worked. Either they detect single features in the environment, or they detect a few combinations of features that stood for a single thing. But in conducting these studies, we found that there was neurons that didn't fit this mold. There were neurons that we termed mixed selectivity. These are neurons that didn't just respond to a single feature or a combination of a few features. They were selected for complex nonlinear combinations of many features with no obvious relationship. You study these neurons in different tasks and different contexts, and the neurons seem to be doing different things, respond to different things in different contexts with no obvious relationship between them. And they also were multifunctional. We used to think back in the 20th century that neurons did one thing. You figure out what each neuron did, you would know what that neuron did. You could figure out the whole brain by figuring out the properties of neurons one at a time. But now we know these many neurons in the cortex have mixed selectivity and they're multifunctional. They don't just do one function. They're like utility players in a baseball team. They do different things in different contexts. So why are these mixed selectivity neurons so important for absorbing all this top-down information? Well, when first Amy talked about the frontal lobe conference, when John Duncan and I first in 2000 presented this idea that neurons are multifunctional at the frontal lobe conference in 2000 in Toronto, we got accused of turning the cortex into a bowl of porridge, that we're just muddying the waters, not making sense of things. And at first it was a concept that many people hadn't talked about, but enough people started collecting more and more data. We collected more and more data, and then you can't deny it. There's these neurons, these multifunctional neurons in cortex. But more recently, Stefano Fusi and Matteo Ragati, working with Melissa Warden, showed computationally why these mixed selectivity neurons are so important for high-level cognition. They add horsepower to the brain. These mixed selectivity neurons are like a neural bazaar where a wide range of information can intermix and be combined. And that makes the brain smarter. It allows the brain to represent things in a high-dimensional representational space. So it could represent more complex information than it can with simple neurons that only have one function alone. And for mathy reasons I won't get into it, by distributing the information across many neurons, it greatly increases the storage capacity of cortical networks. And because of all this information is intermingling together in the same networks of neurons, it allows the brain to learn faster, because all the information the brain needs is right at its fingertips. Just grab the information, put it together. And it also allows cognitive flexibility for the same reason. So computationally, we know now that we need these mixed selectivity multifunctional neurons. But mixed selectivity requires a new way to think about the brain. Because as I mentioned, we used to think that every neuron has one function and one function only. And we used to think anatomy is destiny. Neurons combine their signals because they're physically wired together. If you make new connections, you strengthen or weaken connections, you're changing how neurons combine their signals, and anatomy was everything. That's all that was what determined neural properties. And the idea was that every perception, thought, and action had an anatomically unique network, something we called an ensemble. So I've represented that here by here's one thought, we'll call it the blue thought, and there's one network or ensemble corresponding to that thought. And here's another thought, we'll call it the red thought. And you could develop associations between these things like dinner bell and dinner, and there'll be links between them. But largely, every perception, thought, and action was thought to have a unique network all in of itself. But mixed selectivity means that this is not the way things work. It means that neurons are actually not members of one network, but they're members of multi-networks. They're members, each neuron is a member of multiple ensembles. And I've illustrated that here by showing here's now our two red and blue networks combined together into one network, and they share some of these common elements, these mixed selectivity multifunctional neurons. But if anatomy is destiny, how does this work? I mean, this is what must be the case if mixed selectivity is a real thing, because it means that neurons participate in multiple ensembles. So how does this work? Why don't all the networks activate one another and create a jumble of thoughts? Let's say I want to think the red thought. Well, I could activate the red elements, and then the activity runs in the mixed selectivity neurons. Then over to the blue network, now you have the red and blue networks both activated, and your brain your thoughts are a jumble of mush. So how does this work? How does the brain actually pull a unique ensemble, a unique thought, a unique perception out of this inextricably intertwined networks? Well, one solution that many of us have come to is brainwaves are the solution. Brainwaves are an emergent property. And this is what I talked about earlier when I referred to how now we can study things at the level of emergent properties. We only could do that by studying all these neurons together at the same time. And here's an example. So what brainwaves are, they're the synchronized activity of millions of neurons, a large-scale organization of neural activity. So here's a crowd doing the wave. The wave is moving from left to right. Now, there's no sophisticated apparatus like telling this crowd how to do the wave. This crowd is self-organizing. These individuals are self-organizing based on a simple rule, like stand up when the person on the left of me stands up. Now, if a stadium crowd can self-organize using a simple rule like that, imagine what your brain can do with that principle. And sure enough, we need large-scale organization for top-down executive control. We can't see this level of organization on the level of individual neurons. Think about if I would study this crowd the way we used to study the brain back in the 20th century, I would give a microphone to an individual member of this crowd. And I would hear the detailed conversation. I would hear them yell once in a while, but I would have no idea this wave is going on until I study a bunch of the people in the crowd simultaneously. And this kind of level of organization where you can organize subsets of crowds is just the kind of organization you need for top-down executive control. You're not going to have top-down guidance of thought and action, emotions. You need to control things on the level of crowds, not on the level of individual neurons. Because executing goal-directed behavior requires getting large bits of your cortex on the same page, coordinated and directed towards the task at hand. This kind of level of mesoscale organization. But also, there's something called representational drift. Neurons are fickle, they come and go. Anybody who's recorded from neurons and cortex during complex behaviors will tell you that you record from 100 neurons, 1,000 neurons, and do 100 or 1,000 trials in a row, and you will get 100 or 1,000 different patterns of activity. Neurons fire on some trials, other trials they don't fire. Sometimes they fire here, so they fire there. Neurons come and go. The brain is like the world's largest orchestra. And the cortex, this large orchestra, is playing the song, but individual players are wandering in and out of the orchestra. They're coming in, they're playing for a little bit, they're leaving, they're coming in again, playing a little bit. So the individual players are wandering in and out. So there must be some organization at this higher level of the song, of what the orchestra is playing, and not just the individual players. And you could take this, recently Dimitris Panosas has taken this, he's taken not only the spikes and LFPs, but he's taken it up to a next level up, the electrical field level. Now, I'm not talking about electrical fields outside your brain or even 3 inches away from your brain. The near electric fields hovering around all the brain's, anatomy, all the processes. You can actually measure the contents of working memory from not just the LFPs, but from the electrical fields near the brain. And he showed this in a recent paper last year. And what's cool about it is that you get little or no representational drift. At this level, there's a pure information signal with no representational drift at all. It's just constant signal. It's a level of the song, if I may go back to that orchestra analogy. So, doesn't this sound like a useful level for organizing brain activity? And isn't it easier to control a stable representation? If you want top-down executive control, if you don't control things on this level, what are you going to do? You can control a gazillion synapses on a gazillion neurons simultaneously, and this representational drift is going on. It doesn't seem impossibly complex to control the brain, top-down control at the level of individual neurons. You want to control at the level of this mesoscale organization. That's what brain waves are good for. So the way to think about this is neurons that hum together temporarily wire together. So brain anatomy is not destiny. Brain anatomy is like possibility. Brain anatomy is like the road and highway system. It just says where traffic, neural signals could go. But your thoughts are where the traffic is actually flowing from moment to moment. And that's what we think brainwave patterns do. They're directing the traffic on this possibility anatomy, road and highway system of the brain's anatomy. Now, is there any evidence for this? Of course there is, or I wouldn't be talking about it. So here's one of the first studies of this was when Tim Bushman was in the laboratory. He's now a professor at Princeton. And what he did is he had the animal, this monkey, switch back and forth between 2 different task rules. And what's shown here is a view of the lateral prefrontal cortex. Here's posterior, anterior, dorsal, ventral. Here's the principal sulcus. Here's a bunch of recording sites where we had our electrode arrays. And the colored lines connect recording sites that showed a significant increase in LFP coherence, local field potential coherence, when the animal was following one rule versus the other rule. And as you can see, there's different patterns of this coherence for one rule versus the other, as if the patterns of LFP coherence are actually forming the ensembles. The one rule ensemble for when the animal's following one rule, and then switching to another rule ensemble when the animal's following the other rule. Now, we studied this for all the frequencies from 1 hertz up to 100 hertz or more. And we found that these patterns of coherence for these rules were limited to the beta band. Sometimes it drops down to alpha, so I'll talk a little bit about alpha too, but the alpha beta band from around 10 hertz to around 25 or 30 hertz or so, we see these humming forms, these ensembles for the task rules. And we found these effects for a wide variety of top-down information. Once we found this effect, we went back and looked over other data we collected. We collected new data. Every time we look for top-down information, we're finding it in these pattern forms of beta coherence at this LFP level. So, having said that, let's now turn to studies of how top-down control of working memory can be accomplished by beta rhythms. So Michael Lundqvist came to the laboratory. He started studying, he had a model of how this works, and he started studying data we collected from previously, where the task was a working memory task. The animal just had to hold 2 pictures, we'll call them S1 and S2, in working memory just for a second. And what we did that was unique up to that point is we started looking at neural activity in real time on single trials. Most of the history of neurophysiology is you record 25 trials in a row, then you average across the trials and try to pull out the signal and get rid of the noise. But what if some of that noise isn't actually noise? What if it's a real signal? So we started studying neural activity in real time. And here's what we found. This is now recording. This is local field potentials, power. This is time along the x-axis. So the animal had to hold 2 pictures in mind. The animal saw picture 1, S1, gap in time, picture 2, had to hold both pictures in memory over this working memory delay. And the color is the power of the local field potentials, with red being higher power, blue being lower power, and the y-axis is the frequency. So the first thing that Michael noticed, and this wasn't just a shot in the dark here, this was he had a biophysical model of how to hold multiple things in working memory, and the model made predictions that bore out in the data. So the model came first, and we discovered all this in the data subsequently. And what Michael found was that spiking that was encoding this 2 stimuli in working memory and carrying the memory of the 2 stimuli in working memory were carried during bursts of gamma activity. So you see that throughout the trial here, when the animal is encoding the stimuli, and then periodically during the delay, you get these short, narrow band bursts of gamma. And we found that the spiking that was carrying the information in working memory was coincident in time and space with these bursts of gamma. So that's what's shown here. What's plotted here on the y-axis, now there's an average across trials, not individual trials. But this on the y-axis is not spike rate. It's stimulus information about the 2 stimuli the monkey was holding in working memory, stimulus information from the spike rate. And again, time along the x-axis. 2 stimuli, working memory delay. And the red shows the information about the stimuli gleaned from spiking when we looked at spiking that was occurring during the gamma bursts. And the blue shows what happens when we looked at spiking outside the gamma bursts. Now, the average spike rate was a little higher during the gamma burst than outside the gamma burst, but it wasn't that far off. They were similar to one another. But all the information about the stimuli is being carried during the gamma bursts, and there's virtually no stimulus information into spiking outside the gamma bursts. Now, we also noticed that there was beta bursts, and the beta bursts tend to alternate with the gamma bursts. So you see here's 2 bursts of beta down here around 30 hertz and below, and these beta bursts tend to happen when the gamma bursts weren't happening. So now, again, here's the average across trials. This on the y-axis is the burst rate, with the reddish color being the burst rate in gamma, and the brownish color being the burst rate of beta. And these are, again, LFPs. And we see, look at the gamma. We see the gamma is doing exactly what we've seen a million times before when it comes to spiking in working memory. The gamma burst is going up like spiking does when the stimuli are presented. Here with S1 and S2. Then over the working memory delay, the gamma activity, like spiking activity, first drops down, then it gradually ramps up over the delay. Okay, so gamma is pretty much tracking what we're used to seeing with spiking in working memory. But the beta, notice the beta is doing the exact opposite. Every time gamma goes up, beta goes down. Beta ramps up in the first part of delay, then ramps down as gamma is ramping up. So beta and gamma are anti-correlated. Now, if gamma and spiking is carrying the working memories, what is beta doing? Well, you may remember that we found that patterns of beta are carrying the task rules. They're carrying the top-down information. These actually patterns of beta carrying the rules the animal's actually using to solve the task. How to do the working memory task. So remember, beta and gamma are anticorrelated. So this suggested to us that top-down information is carried by patterns of beta. That's the control signal in the brain, and that they inhibit, if you will, they reduce spiking in gamma, the contents of the brain is working on, wherever they happen to land in cortex. Okay. Now, I won't have time to go into it in great detail, but we recently published a paper like 2 or 3 weeks ago showing how this works for top-down control. The idea is something that we call spatial computing. Spatial computing simply says that top-down control, the brain applies rhythms, these patterns of beta, to physical patches of cortex to selectively control just the right neurons to do the right things at the right time. So the way to think about these patterns of beta, these top-down beta patterns, is beta is like a photographic negative of where you want higher cortical spiking activity. That means higher cortical activity, gamma and spiking. Now, the beta isn't creating more spiking or more gamma where it doesn't land. It's just allowing gamma and beta to be expressed if something comes in and activates the system. But beta is like a photographic negative of where you don't want that information to be expressed. And that's how the brain does computation, we think. So, when Andre Bastos and Roman Lunas were in the laboratory, along with Alex Major, who's taking on the project in the lab now, they decided to study the local circuitry that allows these beta and gamma interactions. And what they looked at is they looked at the activity across different layers of cortex. So here's, we use these laminar electrodes. Here's a laminar electrode with a bunch of contacts along the shaft. Allows us to record from all layers of cortex simultaneously. So why do they care about this layers of cortex? Well, we know anatomically that the superficial layers of cortex are the layers of cortex that feed forward information, sensory information from the back of the brain, bottom up information from the back of the brain to the front of the brain. So if gamma is feeding forward sensory information, it should be stronger in the superficial layers of cortex. And the deep layers of cortex are the feedback layers that carry top-down information, feedback from the front of the brain back to the posterior cortex. And if the beta is carrying top-down feedback signals, beta should be stronger in the deep layers of cortex. Well, that's exactly what they found. They did Granger's causality measures, and what they found in these series of studies is illustrated here by a missing slide. So I'll talk you through it. The idea here is that top-down information is carried in the deep layers of cortex by these patterns of beta. The patterns of beta in deep layers of cortex regulate weaker beta in superficial layer cortex. And superficial layer beta cortex has this push-pull anticorrelated relationship with gamma and spiking, so it allows superficial layer beta to control or gate when gamma and spiking is expressed. And this is how you gate information out of working memory, how you clear out working memory, how working memory is controlled. But when we were doing these studies, we started to review literature and we realized that these beta and gamma dynamics are pretty much everywhere in cortex. You see it here in the motor system. You go back in the visual system, the beta drops down to alpha range, but still bridges a little bit in beta. So we thought, well, maybe this is something that's everywhere in cortex. So we did a series of studies, including this one here that's now in the pipeline, where we recorded with laminar electrodes in 14 different cortical areas, and we found this beta gamma, beta deep layer regulating gamma superficial layer. We found these dynamics everywhere in cortex. We looked everywhere. So we thought, well, it can't just be working memory in prefrontal cortex. It must be something that the cortex is doing in general. So we decided to turn to a more general cortical function. And the function we turned to is predictive coding. Now, predictive coding is a fundamental cortical function because your brain can't possibly process all the information that's flooding into it. It would be overloaded. So what your brain does predictive coding. Your brain is constantly generating predictions of the sensory inputs expected in the next second or so. And now, why does your brain generate these predictions? Well, predicted inputs are not informative. Your brain saw them coming. Your brain was able to predict them. They're not really telling the brain anything new, so they're suppressed and not fully processed. But unexpected inputs, prediction errors, things that the brain didn't anticipate, change, when things change, they're really informative. So they're fully processed, they're fed forward to update our mental model of the world. Now, everybody agrees that the brain does predictive coding, but we still don't quite know how the brain actually does it. Now, one common hypothesis is your brain has a bunch of comparative circuits at every level of processing. And these comparative circuits are comparing the predictions to the incoming sensory inputs, and there's a mismatch that gets passed forward. So that's a fine model, but there's not a lot of direct evidence for that. And there's not direct evidence for these different comparative circuits at every level of cortex. But having our beta gamma results, we thought maybe there's a simpler explanation, a more parsimonious explanation, which we call predictive routing. And the idea is that these patterns of top-down beta are carrying the predictions. The top-down beta is carrying the predictions of a forthcoming predicted visual stimulus or any stimulus. And the beta suppresses the bottom-up gamma spiking that would be elicited by the stimuli and that would otherwise feed forward predicted input. So these beta patterns coming back, and everywhere they land in sensory cortex are saying, you're not allowed to spike. And that's how predicted stimuli are suppressed. And that means that prediction errors, unpredicted stimuli, change and stuff like that are fed forward by gamma that was unaffected by beta, patches of cortex where the beta didn't land. So in collaboration with Nancy Capel's laboratory, we studied this in the cortex of a couple monkeys as the monkeys viewed objects. They're performing a working memory task, but they were viewing objects that were either completely predictable, the same object multiple trials in a row, or they were unpredictable. There was a number of objects and the monkey couldn't tell which one we were going to use. Now what this shows is we recorded from 5 cortical areas. Here's frontal eye fields, prefrontal cortex. Here's the parietal cortex, 7A and LIP. And here's our area in the visual cortex, V4. Now what the red shows is stronger coherence when the animal was performing trials where the object was unpredictable, where all prediction errors. And the blue shows stronger LFP coherence when the animal was performing trials in which the objects were completely predictable. The line shows the pattern of coherence, and the thickness of the line shows the degree of coherence, with the boxes showing the inner area coherence. So what happens here? If you focus on the blue, you see when the animal is viewing predicted stimuli and the brain's generating lots of predictions about what these stimuli will be, you see that blue alpha beta dominate the coherence picture in cortex. 7A is a bit of an exception to most rules in cortex, so we see it stands out. But generally, when the animal is in a situation where the brain can generate predictions and use those predictions, we see that alpha beta coherence is dominating in cortex, because we think that that's the signals, that's the rhythms that are carrying the predictions. And when the animal is viewing stimuli that are unpredicted, that the prediction errors, we see that gamma is dominating cortex, as we would predicted from gamma feeding forward prediction errors, feeding forward bottom-up information. There are prediction errors. Now, Granger's causality showed that, as we would expect, that the gamma coherence, which we think is feeding forward prediction errors, was stronger in the feed-forward direction from visual cortex up to higher cortex. And the alpha-beta coherence that we think was carrying feedback predictions, the predictions about stimuli, they were stronger in the feedback direction from high-level cortex back to visual cortex. And importantly, these LFP effects were stimulus-specific. They were greater in the recording sites in visual cortex, where the spiking preferred that specific predictable or an unpredictable object. So I mentioned these patterns of beta are carrying top-down information. In this case, the pattern of beta is reflecting the actual stimulus that's predicting, and it's targeting the circuits back in sensory cortex that would respond to that stimulus when that stimulus appears. And when the beta is hitting that patch of cortex, the neurons don't respond because that prediction is suppressing that feeding forward of that stimulus. Excuse me. So, up till now, I've talked about the rhythms underlying working memory. I talked about beta gamma rhythms. When I first introduced this topic of working memory, I talked about how we looked in real time on single trials. And we found very complex dynamics, this push-pull, periodic gamma and beta and the spiking. But ever since I mentioned this at the start of the working memory section, I've showed you average data. Well, we have begun looking closer at these real-time dynamics. And one thing we found is that these oscillations in the prefrontal cortex form traveling waves. So here's an example of this is an array over the lateral prefrontal cortex. It's from one subject. It's a single trial of the animal performing a working memory task. And what it shows is these are voltage signals. And as you can see, these waves these oscillations, they don't just they're not standing waves. They don't just go up and down in place like a jump rope. Rather, they move, and they move across cortex. And in this case, they rotate. So this wave is actually rotating around the prefrontal cortex. Okay? Now, what's the big deal about traveling waves? Who cares about traveling waves? Why is that a big deal? Well, traveling waves are super helpful because they buffer information about time and recent network activity. Just having traveling waves gives the brain all sorts of information that it needs for computation. So the way to think about that is, let's say I took a video of a pond and I dropped 4 pebbles in the pond one at a time. I now can take a still photograph from that video, show it to you, and you could tell me how many pebbles have dropped, where they dropped, and how long ago they dropped. And that's what traveling waves buy you. They buy you recent network activity, and not only it gives you recent pattern of network activity and elapsed time. And these are very precise. These waves travel in proportion to their frequency. So alpha waves travel at a certain speed, beta waves travel at a certain speed. We don't really see traveling waves in the gamma and the high frequencies, gamma and above. It's all beta and below, but they travel in a speed proportional to their frequency, which means you can also calculate exact elapsed time. So all sorts of useful information. And we find that these neurons, these traveling waves, in the cortex, they don't just bounce around like billiard balls bouncing off randomly from one another. Much like that crowd doing the wave across the stadium, they travel in very specific pathways. So here's this same subject here. This is the left prefrontal cortex, posterior on the right, anterior on the left. Here's theta, alpha, and beta waves. This is a dorsal ventral. Principal sulcus is the dorsal lateral prefrontal cortex. And you can see that in this case, the waves are not just bouncing around randomly in the prefrontal cortex, they're traveling along a very specific axis, this anterior ventral dorsal, anterior posterior dorsal ventral axis. So just right there, just the fact that they're traveling in a very specific pathway tells you there's something organized going on here. And in fact, they not only travel in very specific ways, they change direction with cognitive demands, especially notably the beta waves. So here's the wave from the animal performing the working memory task. This is the blue shows. So we arbitrarily designated these waves either positive or negative. This could be one or the other, A or B. It doesn't make a difference. And we see, well, in this particular subject, when the animal is performing the task, before the task starts, you get equal numbers of positive and negative rotating waves. So the animal is waiting to start the task, and the waves are just going back and forth like this across the prefrontal cortex. Then as soon as the animal starts the task, one of the waves, the negative waves, they're suppressed, and the waves just start flowing in one direction only. And that is true throughout the entire trial until the animal makes its response. Then you go back to this bidirectional waving thing. Now, why are the waves traveling in that one particular direction? What is it about the working memory task that requires this wave? I'll talk a little more about functions about the waves in a moment, but the short answer is that we don't know now. This is a brand new discovery. We now need to look more computationally at what exactly these waves are doing in the context of this working memory task. But we do see that there are waves in cortex, just like have been seen in visual cortex, just like you see in the retina, and they're very specific, and they change their direction dramatically with task demands. Okay, now last thing I'll tell you about is a series of studies where, in collaboration with Emery Brown's laboratory, we're studying general anesthesia. Now, Emery Brown is a neuroscientist and he's an anesthesiologist. He's interested in knowing more about what makes people unconscious. I'm more interested in consciousness, and we haven't, Emery Brown, you have a joke. We're studying unconscious. I didn't know we're studying consciousness. But anyway, whatever we're studying how general anesthesia makes you unconscious. And there was this test that, it may be disturbing to know, even because anesthesia has been around for like over 100 25 years or so, that no one really knows why it makes you unconscious. Doctors just know it does. And there's been this tacit assumption that it kind of just turns off your cortex. Well, it's not what happens. So here's LFPs and spikes when the monkey was awake. And this is superior temporal gyrus, auditory cortex, posterior parietal cortex, the prefrontal cortex, and frontal eye fields. So these are LFPs on the top and spikes on the bottom. And this is in real time here, so there's about 20 seconds of time here. And during the awake state, you get a lot of high frequency, low amplitude chatter because there's a lot of waves, waves synchronizing and unsynchronizing. Thoughts are happening moment to moment. So a lot of complex, humming going on. And you get a lot of high frequency, we'll call it FM radio chatter. But then we anesthetize the animal with propofol anesthesia, commonly used anesthetic, and it didn't just shut off the cortex, it drastically switched the frequency profile of the cortex. So all that high frequency FM radio chatter was replaced by this low frequency delta hum. So you see here's the LFPs. You can see now there's this low frequency component about 1 time a second. And the spiking in all these cortical areas, they cohere, they line up, they synchronize to this low frequency delta hum. So instead of this high frequency chatter we normally associate with cognition and consciousness is replaced by this low frequency hum that seems to disrupts cortical communication. And to further test this hypothesis, we decided to see if we can wake the animal up from anesthesia by restoring high-frequency signals to the cortex. And we did so by high-frequency electrical stimulation of the thalamus. And we started high-frequency stimulating the thalamus, and the cortex went from this state, this low-frequency state, back to this high-frequency state. And the animal wakes up, opens his eyes, starts moving around, and then you turn off the electrical stimulation, and for about a few seconds, the animal's still awake, then it goes back down to unconsciousness, and the brain goes back to this low frequency state. So we know that that anesthesia seems to cause loss of consciousness by switching to this low frequency state. Well, that's curious because your brain also switches to delta, the exact same frequency when you sleep. You go into slow wave sleep when you sleep. But sleep is very different from anesthesia. In sleep, you have a sense of time has passed. If you sleep for 10 hours, you wake up thinking you've been away for a long time. And also, I could easily wake you up from sleep. And third, sleep is memory consolidation. You consolidate memories when you sleep. Now, the opposite is anesthesia, where you have no sense of time passage. I can't wake you up. And importantly, anesthesia causes amnesia. You get amnesia for the minutes up to when the anesthesia was delivered. So why is sleep so different from anesthesia when at least slow wave sleep and anesthesia look pretty much the same, at least from the frequency domain? Well, we found out it's because anesthesia changes it doesn't just change the frequency component, it changes how the waves travel. So traveling waves, I mentioned that we talk a little about function of traveling waves. Traveling waves are very useful for memory consolidation. They can create precise timing relationships that can induce spike time-independent plasticity. If two neurons, if they spike in a certain window, they will strengthen their synapse. And if they fire the long relationship, the A-causal relationship, you'll get LTD, they'll decrease their synapse. All this takes place within a very narrow time window. Well, that's what traveling waves can do. So if you have like a delta wave, for example, the delta wave I'm talking about, where they're rotating across the cortex, right? They're rotating at a certain speed. Well, all along that axis of rotation, you're getting these timing relationships in spiking because you're inducing this excitability. So how the neurons fire in relation to one another will depend on their position along the axis. If they're near the origin of the rotation, they'll be very quick, and it slows down as they move along the axis of rotation. So just by moving the center of the rotation along a network, you can induce synaptic plasticity, spike time independent plasticity, anywhere you want to along that network. So, and sure enough, rotating delta waves during sleep spindles have been associated with memory consolidation. If the delta waves don't rotate, you don't see rotating delta waves, you get poor memory consolidation after sleep. So there's something about these rotating waves that are very useful for plasticity. Well, what we found is that propofol anesthesia actually unrotates the delta waves. So there was two types of waves we could detect: planar waves just moving straight across the array in a straight line, or rotating waves that are rotating across the array. And when the animal's awake, which is shown here, this is a fraction of the waves either moving planar, just straight, or rotating in blue. Out here is when on the left here is when the animal is fully awake. Now we induce propofol and the animal loses consciousness here at LOC. We turn off the propofol here, these are minutes now, and then the animal recovers consciousness, partially recovers consciousness. You don't really wake up fully from anesthesia for a little while, but the animal starts recovering consciousness around here. And as you can see, when the animal's awake, half the waves we classify, the delta waves we classify as just planar, and half of them are rotating. Then the moment the propofol starts, the rotating waves stop rotating until they drop, they're pretty much out of the population. All the waves and delta waves now unrotated, and now it's all planar waves. And then as the animal recovers consciousness, you get the rotational component back in. So we think this might explain why anesthesia induces amnesia and sleep does the opposite. Because if propofol anesthesia is unrotating delta waves, you don't get that spike time and the plasticity, you don't get the memory consolidation, and you have amnesia not just for when the drug was taking effect, but for the few minutes leading up to the drug. Okay, and now finally, last thing I'll mention is that one thing I get asked, and one thing occurred to us is, how do the brain maintain working memory between the waves of activity? I showed you this sparse activity. There was these bursts of gamma and spiking, and with gaps between them, and this is all and these waves are moving across cortex. Well, waves moving and gamma bursts happening occasionally mean there's times when the neurons aren't spiking at all. So Leo Kostakov and Adam Eisen, they building working memory models where they've been building in short-term synaptic plasticity. This short-term synaptic plasticity mechanism is one of Paco Marquish's last papers. I think it was Wong and Goldman Marquish, where they discovered this short-term synaptic plasticity mechanism in slices from the prefrontal cortex. So they've been building these mechanisms into models of working memory, and they found they could build models of working memory that do working memory just fine when there's this sparse firing, these complex waves, and the synaptic plasticity, the short-term synaptic plasticity is carrying the information between the bouts of spiking. But that wasn't too surprising. If you put plasticity in the network, it'll remember, right? Well, what they found is there's also these side benefits from short-term synaptic plasticity that have nothing to do with working memory per se. They find that these short-term synaptic plasticity also makes neural networks work better. It makes them more robust to noise. You can make the signal really noisy and the network does just fine. Whereas you have a network that does persistent activity alone, it's very subject to noise. It's more robust to degradation. If you have in their networks when there's no synaptic plasticity, if you delete as few as 10% of the neurons in these units in these models, the units just completely fall apart and can't do the job. But if you have this short-term synaptic plasticity, you can randomly delete 40% of the synapses in the models, and the models still perform perfectly fine. So you get something called graceful degradation, which is very important for brain networks. And finally, it helps stabilize networks so the neural activity doesn't run wild. If a highly interactive connected systems like the brain will tend towards chaos, so you'll get chaotic activity. But the short-term synaptic plasticity, along with a couple other mechanisms, stabilize networks so you get nice focused neural activity, and the brain can do the same computation again and again and again without running wild. So in sum, the neural activity underlying cognition is not smooth or steady state. It's complex, dynamic, and rhythmic. There's emergent properties like oscillations and traveling waves that allow the brain to self-organize, organize activity on a scale that's useful for computation and useful for cognition. Low frequency alpha beta waves are the control signals, the pattern control signals, and they control the higher frequency gamma waves and the spiking that carry the contents of thought, perceptions, and working memories. Now, I don't want you to think about alpha, beta, or gamma as having a function. It's like saying spikes have a function. They don't have a function. What they are, they're different states of the network. Gamma is the higher energy state of the network where you'll get lots of spiking, and alpha beta is a lower state of the network where you get less spiking. And the way this works is that alpha beta doesn't inhibit gamma per se. If you drive a network down into the alpha beta state, it can't be in the gamma state. So there'll be less energy, less spiking. And if you drive a network up to with an input, like a sensory input, drive up to a high energy state with it goes into gamma, lots of oscillation, lots of spikes cause lots of crossing of the spike threshold, lots of spiking, and you get lots of activity. But if the network's in gamma, it can't be in beta, and if it's in beta, it can't be in gamma. And that's really how it works. It's not that the alpha and beta have different functions or they actually have inhibitory circuits or something. They're just different energy states of the network, and the energy states are useful for different functions. And we think this is a cortical motif. We've seen it in 14 cortical areas now that plays a general role in regulating cortical function, working memory, attention, predictive coding, pretty much anything the cortex does. So I thank you for your attention, and I thank all the students in my lab who actually do all the hard work at making these results possible. Thank you.
Thank you so much, Earl.
Happy to take questions.
We knew this would be thought-provoking, so I'm sure there are lots of questions people want to jump in. And I'm sorry that my husband is printing something right next to my computer.
I don't hear anything.
Maybe I could start quickly. Earl, this is a really very provocative talk. I have just a couple of questions because I don't want to take over. A lot of people have questions probably. Number one, the experiments that you do, the animal is fixating. So there is no eye movement, there is no changes in the retinal image. Is it possible that when you actually do experiments in which there is more naturalistic vision, a lot of these oscillations and a lot of these phenomena that you see actually change? Because the whole cortical dynamics will definitely change as soon as you have corollary discharge and as soon as you have changes in retinal images.
Oh yeah, yeah. You definitely see the cortex will modulate by eye movements. It'll even modulate by microsaccades. So we've done that. We looked at both the microsaccades. We've also looked at. We have a few studies where the animal is doing free viewing of this thin line and make a choice, and we see these same dynamics play out. I mean, once you account for the eye movements, it looks exactly the same. And this should not be surprising. I mean, some of the stuff we see in cognition that we associate with that are non-movement, it actually turns out they have a movement component. Like there's Bob Desmond recent work where he looked at covert attention, and it turns out if you look at the animal's microsaccades, the covert attention, the effects of covert attention are tracking the microsaccades. So the microsaccades are actually kind of driving the effects of covert attention, but that doesn't make it an artifact. I mean, your brain evolved to make movement. Your brain, you move or you die. You either move or you die, or you kind of move to procreate. The brain evolved to make movements. So it doesn't surprise that in the end, a lot of this stuff will be driven by connections in the motor system. Where did cognition come from? Cognition evolved from the systems for voluntary behavior. So everything I'm talking about is modulated by the eye movements, microsaccades and macrosaccades, but in a principled way. So you still see the same effects. It's not being an artifact of how the eyes are moving.
Yeah. My second short question is like oscillations. Usually, for example, if you look at epilepsy, has been when oscillations propagate too much, basically you hurt neural coding and you hurt a state of consciousness. These oscillations that we see in the network, actually they have to remain to a group or to the ensemble that does that. And also, I think that you have been seeing that oscillations may just propagate. And I think in that sense, how do you keep those oscillations constrained to the ensemble and to the parts of the brain that are participating in the encoding?
Right. Well, a great question. But I'll point out that the sentence you just said, I can replace oscillations with spikes, and the same question holds. Spikes go crazy under epilepsy. How do you organize spiking to produce top down? Every question about that when it comes to oscillations is also true for spiking. But I will say that that's the problem we're trying to solve here. If you had a brain that ignored oscillations, just did spiking alone, how do you organize an ensemble on the level of individual neurons? That seems impossibly complicated. On the other hand, networks oscillate, electrical circuits oscillate, neural networks oscillate. It's what they naturally do. So networks probably got complex enough so they started to oscillate, and that oscillations are control, a mesoscale control of neural activity, and evolution probably figured out, well, if I can use this mechanism, I can actually push around the oscillations, control the oscillations, and now have a mechanism for controlling the brain. Now, how that control comes down, well, if I had the answer to that, we'd have the whole brain figured out, and we could all go home. But I will say that, we have done a lot of work where what the prefrontal cortex is doing to these bootstrapping operations with the basal ganglia. It's forming these higher and higher level representations, and what the representations are representations of goal states and the mean to achieve them. We would say that you activate the right pattern of activity in the cortex, the right prefrontal cortex at the right time that corresponds to a goal state and a direction. That sets up this pattern of beta and now tells the rest of the cortex how to engage in that goal-directed behavior.
So it would be fair to say that the way to control these oscillations is connectivity, because that's the way that we relate the spikes. Neuron A is connected to neuron 2, and connectivity in this case is what actually bound those oscillations. Would that be fair to say that?
Don't get me wrong, I don't hate spikes and I don't hate anatomy. You can't do anything about spikes and you can't do anything about anatomy. These oscillations, so again, the anatomy is like the road and highway system, where the thoughts are traveling on them. Well, you don't have anything until you have the road and highway system. Is first of all, it tells you where you can go and how you can go. So it's extremely limiting and controlling to begin with, but that's all latent information. And how the control actually happens is controlling how the neural activity dances on this highway system. And that's what we think the oscillations are doing. And personally, networks naturally oscillate, and I find it really hard to, and as I think I've demonstrated in this talk, is that by looking at these emergent properties, you get all this useful information on this mesoscale level. I find it hard to believe that the brain would evolve and start oscillating, and evolution would somehow take advantage of all this useful information at a very useful level. Thank you.
Hey, Jacob, you have a question?
Yeah, thanks. A long time no see, Earl, from
Hi, Jacob.
last week. I wanted to follow up maybe on a little bit of what Julio mentioned, which is you see like these beta rhythms, especially in the deeper layers. And I'm wondering, like kind of back in the day, you did some striatal kind of learning recordings. And I'm wondering like what you think about that, because I could almost, I guess, similar to what Julio asked, like you could think of beta as kind of this controller, or again, you could sort of flip it and say, like, is there just sort of these corticostriatal loops doing prediction or control or gating? And then sort of beta is the language that connectivity like talks in from striatum to PFC. I'm curious like how you think those loops may be involved, almost analogous to sort of like what Joni's done with hippocampus and OFC, where if you disrupt those two together, they stop coordinating like model building in the world. Like the same thing you could think of striatum and LPFC and working memory. Sure,
that's a great question. And the work from like Sabine Kasser and others have shown that what these subcortical structures like the striatum and the thalamus bring, they give the cortex these lower frequencies like theta and beta, right? So that's being generated. We can't, I'm sure everybody here would agree that we can't think of the prefrontal cortex or cortex alone working in isolation. The cortex is an extension of what was there subcortically before the cortex evolved. So there's all going to be it's all going to be interactive. We got to understand the prefrontal cortex by understanding the basal ganglia and thalamus and the hippocampus and cortex. So yeah, these rhythms are being applied, being largely given to the cortex by these subcortical loops, but then they act on them in the sculpting way. And we did a study where the animal learned new categories, like over the course of a 3-hour recording session, animals learn these new categories. And what we found was that as the animal starts learning the categories, the first thing happened, there's this big general increase in beta coherence between the prefrontal cortex and the striatum. Then as the animal's learning the categories, that big increase in beta in general then starts resolving itself to two different patterns of beta for the two different categories. So we actually see these patterns of beta evolving between the prefrontal cortex and striatum during the learning process. And when you see that, there's seems to be something going on there, obviously, but I wouldn't say it's just a byproduct of the anatomy. It's all part and parcel of the anatomy and the rhythms that they create.
Yeah. I'm curious, have you looked or plan to look for those kind of single trial bursts and striatal signals at all?
Yeah, we find them there too. Yeah, mainly, we both the beta and gamma. We can see them both in striatum as well.
Guillermo?
Yeah. Thanks. Can you hear me?
Yes.
Thanks for the talk. That was really, really good. I have two questions that are kind of unrelated, but the first one is regarding the example that you show to illustrate the waves, the people in the stadium that they stand up all together. In that example, the all the activity is actually local, right? So there's no real propagation of activity. It's just local activity that is in synchrony. So do you have any evidence that in the traveling waves case, when you measure in the monkey cortex, that's not all the same, that the case of the stadium people? That so to some extent, my question is like, do you have any evidence that the actual waves physically propagate, or it's just a local synchrony kind of phenomenon, right?
Right. Well, what's propagating are electrical. So nothing's actually moving. It's the change in electrical. That's the change in electrical voltage that's moving, right? So it's nothing's actually moving. It's the electrical activity across the cortex that's moving. But your question is, yeah, it starts out locally, but that's exactly what you want. So you're building a brain that where local interactions can then lead to a global organization. Then you control the global organization. Now, how is that controlled? Well, neuromodulation can actually take place in a very precise way, and that's going to control excitability. So now you have a mechanism now where you can actually take something the brain's ordinarily doing, creating these waves, and the waves are moving waves, and you can get the waves by just linking together a bunch of coupled oscillators, and you have that, and now you have a little neuromodulation there. Now you have a system that's going to oscillate anyway, and now you have ways to control the oscillation. Now you have ways of controlling this scale of excitability. So I'm sorry, did I answer your question?
Yeah, I think so. So probably it was just. And my second question was regarding your proposal of the role of short-term plasticity.
Yeah.
Is that particularly short-term potentiation or depression, or a combination of both? I didn't get that part of the plasticity part of the story. Yeah.
Yeah, it was pretty quick. So from the Wong and Goldman and Kish paper, it's calcium dynamics that lasts about 750 milliseconds or so, and it's excitability, it's potentiation.
It's a potentiation. Yeah. Okay.
So essentially what's happening is that the network is spiking, then the spiking leaves an impression in the synapses for a little under a second.
So is potentiation of the excitatory connections between pyramidal cells locally?
That is correct. Now, whether there's probably more than one mechanism at play here until we get down to that level of we won't be able to sort them out. But this is a computational model based on the short-term synaptic plasticity mechanisms that are in the literature.
Yeah. Okay.
And
thanks.
Fit with all the magnified calcium we see in those layer 3 spines.
Yes. Thank you.
John, you have a question?
Yes, thank you. Thank you for the wonderful talks. So, while reading the literature, I find it seems like visual cortex is more have like a stronger gamma, while the other auditory or motor signal module maybe more stronger beta. When you're talking about this top-down, bottom-up control or prediction, you are really thinking about specific frequency matters, or you just want difference in two rhythms. And also, gamma is a large range, right? So you come from 40 to hertz. So what's your takeaway from that?
That's a great question and a great point. We have to remember these labels, alpha, beta, theta, gamma. They're made up by somebody like 150 years ago looking at the very first EEG traces. They're arbitrary boundaries. So, there's not any sharp boundary between alpha and beta, or the gamma and beta. There are different differences of frequency. I don't think the exact frequency matters so much. And, in the individual brain, sometimes in some monkeys, you'll see the beta is higher than in other monkeys, or the gamma range is higher. So I think what's most important is the difference in frequencies, the low versus high frequency. We think in general, the high frequency, and again, there's a different energy states of the network. So gamma is associated with high energy, and that's good for processing inputs as they're coming in, and it's also good for creating motor output. And lower frequencies are incompatible. They're lower energy states. So if you want to stop sensory information from being processed, you want to stop prevent a motor output, you drive the system down to the low energy state, and then nothing happens because there's less spiking. And classic example from the motor system, you record from motor cortex and you tell the person or the animal, press this button, but wait, don't do it, wait till I say go. And the whole time they're waiting, beta, 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 beta. And the moment you get the go signal, beta drops, gamma goes up, and the response happens. And we have a study we published in 2018, I think, in Nature Communications, where we actually had the animal do this complex working memory task. The animal has to gate information out of working memory. And you see that exact same thing happen, this beta gamma cycling happening in a permissive way during working memory. It looks like motor cortex, except the animal's not gating a motor response. The animal's gating information in and out of working memory. So we think it's the same mechanisms that motor control. It's the same thing that's happening in cortex. But the exact frequencies don't matter as much as the difference in frequencies, to give you the short answer to your question, I think.
So Earl, from the perspective of the single neuron, which is where we sit, you're up there in the satellite and we're down sitting next to the trees. Could you put up your last slide?
Okay.
Because I think it's helpful to talk about it with it was a model of layer
Yep.
3 and 5, the one before that. So
Yep.
it'd be your last data slide, or 29 is the number. Yeah. So two things. One, at least this is now for just dorsolateral prefrontal. There's a lot of evidence, very direct, that layer 3 is also part of the feedback, for example, to parietal. Very much so.
Yep.
And so I can see where the bottom up, top down framework would fit for earlier, but that up here in prefrontal, I think it's more complex than that. And that the deep layers, your own work would suggest, as well as others, a lot of this is related to response part of the task. And for example, both Pat and Min's data, we would add to this that the layer 3 circuits during the delay, which is when you get the persistent firing and you yourself get gamma, there's evidence that the response-related cells, which we think are in layer 5, consistent with your data, because they're the ones that are sensitive to dopamine D2, they get inhibited during the delay. And so it suggests that, and this could be very much in addition to, that the layer 3 cells are actually inhibiting the deeper ones during the maintenance in working memory.
Sure.
And that this back and forth relationship actually is at least 2 ways as part of the microcircuits, at least up in prefrontal.
Yeah, I agree. This is biology, and it's the brain. You know, it's not going to be as simple as superficial as this and deep layers that. There's a lot of truth to that, but there's a lot of crosstalk between the different layers too, and it's certainly going to be more complex than superficial versus deep. I mean, absolutely. This is the brain. This is a model. It's just a simple stand-in for an idea, and then when we flesh out all the details, we're going to find out, of course, as we always do, that everything is way more complex than we think it is, especially when it comes to the brain.
And it seems like a particular challenge of making causal manipulations to test these ideas. Like the propofol is a kind of crude hammer, but to get more delicate about it, how do you actually take out a particular frequency to see what it does to cognitive operations, for example?
Okay, let me ask that question 2 ways. First of all, what we're doing is we're just now starting a series of studies where we're going to be doing closed loop electrical stimulation. Closed loop electrical stimulation is you read the actual beta signals from the brain, and you either match stimulation to the beta signals or antiphase match, so you can actually turn up and down the volume of the beta signals. So we're going to manipulate the control signal. We just got this.
And from where will you do that?
Where? Everywhere we want, feel like recording. We're gonna.
So you're going to do it globally all at once?
Oh, we'll do it in prefrontal cortex, and then we'll do it in parietal cortex, then we'll do it in both areas.
So sequentially.
Yeah. So what we're going to do is we're going to take those control signals, we're going to manipulate them. And we just got brand new technology developed, just arrived in the lab this week, where we have a new closed loop electrical stimulation system that we developed that has a super fast 4 millisecond read write latency, so we can do any frequency we want. As far as causal manipulations, I'll mention quickly that one study that Alex Major's doing in the laboratories, he's deactivating deep layer prefrontal cortex. And when he deactivates deep layer prefrontal cortex alone, he gets a loss of only prediction signals back in V4. The bottom up signals are fine, but all the prediction signals are gone. So that shows that suggests that the prediction is actually coming from deep layer, and you don't get that from deactivating superficial layer cortex.
I was just going to say that would be an important control there.
Yeah, well, I've been doing this a while. We know about controls. So we've done all that, and we do see that the deep layer prefrontal cortex is in fact providing the predictions. But when we talk about causal manipulations of these oscillations, let me ask you a question: What causal manipulations are there that shows spike rates alone have an effect on brain function?
Mostly that when we do really tiny injections that we know reduce persistent firing, we also get impaired working memory. And we can see, we and others on trial-to-trial basis, that the monkey's making errors on trials when the persistent firing doesn't get across the entire delay.
Yeah, we see that for oscillations. So we had a study where we had to do this complex task and when we looked at the spiking activity, we could tell the animal was going to make a mistake, but we couldn't tell why the animal was going to make a mistake. It was a complex task where the result was yes or no from the animal. But when we looked at the beta gamma, we could tell why the animal was we could tell more about the animal's mistake from looking at the oscillations than we could from the spiking. So that's one thing. But the second thing, you mentioned that stimulating the brain. Anytime you stimulate the brain, you're changing spike rate. You're also changing the local field potentials in the oscillations. So when people ask what's the causal, can you do this? And so it affects the oscillations affecting, but not spike. No, you can't do that because spiking and oscillations, they're all part of the same thing. You affect one, you affect the other. So when people ask,
that was the point of our paper in Jane Neuroscience against you.
Okay.
No, because the way the argument was framed, it was that these things were separate.
If that's the impression you got, I don't think we didn't frame it that way. I'm sorry you got that impression. We never would say something like that. And we said things the opposite of that in like our working memory 2.0 review. We talked about how they're not separate things. They're beta gamma not separate. It's all part and parcel of the same thing. And beta doesn't do one thing; it does multiple things. But the point is, I often get that. Well, if you think oscillations are important, show me a causal manipulation. I turn around saying, show me a causal manipulation that spike rates are doing everything. There is none, because the moment you start injecting current into the brain, you're changing everything. So the quality of evidence for spikes versus oscillations are exactly the same. It's all.
Earl, do you think, to build on that, why there'd be bursts of gamma? So we know here you have this group of neurons with persistent firing across the delay, but you only get a burst of gamma. Is that because? And you would say it's during those bursts that there's the most information.
Yeah,
would you hypothesize that that's because at those moments is when that group of neurons has sufficiently coordinated activity to entrain a larger group enough that you get a burst into the gamma spectrum?
Well, first of all, when you talk about persistent firing, you need to not average across trials, because once you average across trials, you're not.
We don't need to do that. We see it on across in a single neuron on a single trial. You can see it across. So if you had enough of, I would think that, let's say you have 10 neurons with the same preferred direction. And they're not going to be firing at exactly the same time across that delay period, but there may be moments across those many seconds when their coordination is such that it's enough to then entrain neurons around them as well. And that might get then reflected as a burst in gamma, because now you have enough neurons affected to reach that higher level. Does that make sense to you?
Sure. Now, the way it works is in the original Lundquist model, which is that you have all these ensembles all trying to inhibit one another. right? And what those gamma bursts are, they're close to spiking with, you're activating an ensemble. Then as soon as it gets activated, because of lateral inhibition, all the other ensembles go to shut it down. That's why you get short bursts of
So
gamma.
we're saying the
If I
same
may-
thing, but in converse,
Can I
I'm
finish my
saying
sentence?
that you have enough neurons having the same shared persistent firing in coordination to then set up the gamma. So I think we're chicken and egg here.
Well, you let me finish my sentence. Let me finish my sentence.
Okay, sorry.
So the reason why the system is set up with all these short bursts of gamma and then hitting one another, because every one of those gamma bursts is in a attractor state being activated very briefly. Now, what happens when you activate two attractor states at the same time? They smush together, and you can't hold two things in working memory. So what those gamma bursts are, and we're testing this now in our analytics because now we have enough electrodes, are different ensembles being activated. So you want the gamma burst to be short because you want the attractor state to only be there for a brief period of time, and the attractor states, if you have multiple objects of working memory, you want attractor states on at different times so they don't interfere with one another. That's why you get the short. That's why the gamma burst and the spiking is short. You don't want interference between the attractor states.
Oh, may I actually, oh, sorry, because I think I have a suggestion for the, some of our group, what it's doing is looking at intrinsic properties of cells across the cortex. But most of the models that exist in oscillations, they really don't look at intrinsic properties of cells. And each cell, for example, Guillermo found that prefrontal cells, they fire these bursts, and you have this type of bursting cell in the prefrontal cortex layers 2 and 3. But if you go to parietal cortex, you don't have it. They're probably regular spiking, and they don't have that kind of property. If you were to incorporate into the models that have the biophysical properties of the neurons, do you think that it would have any impact in the way that you look at oscillations? So oscillations is not just the matter of the network. You also have to take into account the transfer function of the cell. Some of these cells, they are,
Absolutely. So what's the question?
The question is that if you were to incorporate the biophysical properties, if you were to get biophysical models in which you have real biophysical neurons, the prefrontal neuron is different from the V1 neuron, and it's different from the empty neuron. What would you think that would impact the oscillations that you see?
Well, you need to do that by testing it. But the Lundquist model is a pure biophysical model. That's where it came from. That wasn't built. He didn't build the model to create oscillations and gamma bursts. He built a biophysical model to do working memory, and these gamma bursts and stuff were an emergent property of that network. It was what came out of the biophysical model, but it was a pure biophysical model. Lundquist et al. 2011.
Yeah, no, I know that the biophysical model, I'm referring to Hawking and Huxley equations, differential equations in which you have the different conductances, you have the different things. I don't think that model got to the depth that you will model those neurons that way.
Yeah, well, we can and should model them, but there's the brain is doing it, so you got to figure out why the brain is doing it. And I find it hard to believe that people who say stuff like, "Oh, the brain has to ignore oscillations because it did not factor them out." What is your brain? Your brain evolved a system for oscillating, and then there's a giant Bose noise canceling headphone in there somewhere that cancels out the oscillations. I mean, if the brain is doing it, we have to figure out why the brain's doing it. And you're right, that will be approached by different levels of biophysics, different parts of the brain, because you're right, the prefrontal cortex, parietal cortex, they have different biophysical bedrocks, so you need to figure out how and the oscillations aren't exactly the same, the prefrontal and parietal. I showed you that, where parietal cortex, for example, was an exception to a lot of different cortical areas when it comes to the oscillatory dynamics. So yeah, this is kind of stuff we need to figure out in the course of figuring out the brain. I completely agree, but it's all got to be sorted out. But my philosophy is that there's no epiphenomenon in the brain. None of us know enough about the brain to say anything's an epiphenomenon. I'm going to assume if there's a signal in the brain, it must be useful, and I'm going to measure as many signals as I can until we all figure out the brain.
Yeah.
Guillermo, did you also have a question? I saw your hand up for a bit.
No, it was already answered, but I think that's going back to the debate about spiking versus oscillatory activity. I think in the case of spiking, it's pretty well demonstrated in many cases throughout the nervous system of many animals that, for example, spike rate is very directly related, let's say, with the intensity by which a muscle contracts or something like that. So then unlike oscillations in which I mean, I work on modeling oscillations and looking at biophysical properties of neurons to understand the mechanism of oscillations. Perhaps Amy's question about, well, do we have a demonstration that the oscillations, you perturb them and that you change something about behavior or motor activity or whatever, is a little weaker than for spike rate, in my view, right? So, but I may be wrong, yeah.
Well, first of all, because oscillations aren't, look, if you want to explain the neuromuscular junction, you don't need oscillations. If you want to explain how two neurons in a petri dish together, you don't need oscillations. But that's what we're trying to explain. We're trying to explain cognition. And that's where these emergent properties are going to be most important. And there's also lots and lots and lots of examples of changing the oscillatory dynamics of the brain and affecting behavior, just like there is for spiking. And the recording level, there's the same correlational evidence for, you get everything you said. I can read the contents of working memory. Now, again, I'm not saying spikes aren't important. I love spikes. We study spikes too. We study spikes and we study LFPs. LFPs, they're all important. But I can read a lot of the stuff out of the brain by using LFPs alone, not even looking at oscillations. I'm not saying the spikes aren't important. I'm just saying that they're all important. And I personally, I think when it comes to understanding the brain, a approach, a paradigm, a model that explains lots of phenomenon is better than one that explains one phenomenon and ignores the rest.
I think that I find the whole thing of spikes and LFP a little bit crazy, because the LFPs that we read out is just a result of spikes too. I just want to say we are not anti-oscillations. We're just trying to understand the things that you're proposing and trying to put it within the framework of this diversity of structure that we have in the neocortex, where prefrontal neurons are bigger, they are branched out, they have a different synaptic integration as neurons in V1 or in V4. That's what we're trying to understand.
No, Julio, I've known you long enough. I know that you're very open-minded. I completely agree. This idea of spikes versus the versus, I don't know where that comes from. We're just studying the same thing at different levels of analysis. I don't know why people are hung up on spikes versus LFPs. That's a false distinction right there. It's all the same thing we're looking at. It has to be.
Well, I think
that was recorded, so that'll be wonderful to have to play, because I'm very grateful to hearing you say that, Earl, because I think it's often taken to be one versus the other. Well, in the way the field has come to see this.
Well, maybe I'm partially to blame for that, but I don't think I've ever said that there's different things. And you know how science works, or anything works. Like when someone has an idea or a model, you first make a cartoon of it, then you attack the cartoon. So when I talk about LFPs being important, people say, "Oh, don't you you must hate spikes?" No, that's not it at all. And I've tried to never give the impression that there's a one or the other kind of thing here. I feel quite the opposite. I love spikes. I love anatomy too. I'm not missing that either.
Something that I just want to say shortly is that, Earl, your paradigms have been also different from the paradigms that people use. It reminds me a little bit about the difference between semantic and episodic memory. Your paradigms, it reminds a little bit more of the episodic memory of the prefrontal cortex, like the episodic buffer of Padley, because your paradigms, they have temporal dynamics. And when you have a paradigm where you don't have temporal dynamic, maybe persistent firing is sufficient for doing that, even if with or without oscillations.
I agree. I also think you tend to see more persistent firing if the animal knows the motor response and is waiting to make it. Because it's kind of like that's kind of situation where you're revving the engine, keeping your foot on the brake. So if there's a known motor component during the working memory delay, you'll tend to get higher spiking activity. That's also something to think about.
Tips?
So as an anatomist, if I can ask an anatomy related question. So I was wondering how you sort of think about lamina differences in circuit properties, molecular features between subpopulations of cells, especially thinking about cortical hierarchies and how this plays into dynamics for oscillations.
Yeah, well, again, this is this model, which the level we're working at is superficial versus deep, because in general, that's the feed forward feedback, but it's not quite that simple, of course. But one thing bit of anatomy I would add in related to what Amy or somebody asked me earlier was that, the middle and deep layers of cortex are more strongly connected to the subcortical structures like the thalamus. So that would make sense why you get the lower frequencies in the deep layers of cortex. In the superficial layers, you get more gamma because they're less directly connected to these subcortical structures providing lower frequency, and they're more the feed forward layers that are feed forwarding the high energy sensory signal.
I was really thinking about that a lot when you were talking about projections to thalamus in particular.
Yeah.
And we know prefrontal projects to reticular thalamus throughout almost the whole length.
Yeah.
So you'd have the ability to have these very wide-ranging effects.
I mean, one work I referred to, Sabine Kasser, we just saw Sabine Kasser give a talk at the CNS meeting last week, and she talked about how even sustained attention is not steady. It should be the most persistent thing in the brain, sustained attention. Instead, it waxes and wanes at theta. Well, the animal's just sitting there waiting for a target, and behaviorally, the attention ability to attend waxes and wanes at theta, and that's in synchrony with these LFP theta rhythms in cortex, and that's being driven by theta coming from the thalamus. Okay, so I think that fits in with the subcortical story, but also goes to show you that, look, the brain's going to oscillate. So everything is happening within the context of these dynamics, and they do make a difference, because even when you want to keep steady attention on something, you can't do it steadily. Yet you still have to revert back to an oscillatory dynamic. And we could argue there's a reason for that, because, if you want to pay attention somewhere, you don't want to not sample other areas. Something else may happen along. So maybe that periodic thing is a way of freeing up attention for a moment of exploration,
Would make survival sense, yeah. Are there any other questions? This was so much fun, Earl.
Oh, fun for me. Thanks.
As always,
Thank you.
love how it pushes our thinking.
I try. Charlie Gross taught me that dogma is a thing to tear down. By the way, it's Charlie, not Chuck, just so you know.
Oh, yes. I'm sorry about that. Yes.
No, it's okay. I don't think anybody's ever
things like that are important. Yeah. Anybody else? So a final thank you, then. Take care, everybody.
Good fun.
Thank you very much.
Thank you, Earl. Thank you.